All right, everybody, we're going to get started, and you get to see me talking into my phone for a couple of reasons, but I just wanted to let everybody know, thank you so much for being here as part of the Hatfield Marine Science Day online. We are coming to you live from Oregon State University's Hatfield Marine Science Center located in Newport, Oregon. My name is Cinnamon Moffett, and I'm the Research Program Manager, and I will be your host for today's events. We are really excited to try this new format where we get to take you behind the scenes in our labs. There's a lot of technology going on, so it might be a little bumpy. Be patient with us, but we are excited to show you around the new genetics lab in the Marine Studies building. We are using a webinar format, so your mics, camera, and screen share are turned off, but we hope that you use the question and answer to go ahead and talk to us and put any of your questions in. You can also use the chat if you have any technical issues or questions that you would like to talk to the panelists directly, you can put those in there. I also want to let folks know that we are recording this event, and so it'll be posted on the Marine Science Day web page in a couple of days. So before we get started, I just want to do a quick introduction for our speaker who you can't see yet, but you will in a second. Debbie Steele is a senior faculty research assistant with the Marine Mammal Institute Cetacean Conservation and Genomics Lab at Hatfield Marine Science Center. She has a master's in marine science from the University in New Zealand, and Debbie has been with Oregon State University at Hatfield since 2006. Debbie is responsible for the daily operations of the lab, assists graduate students with molecular research techniques, and curates the tissue and DNA collection under frozen preservation. So Debbie, I'm going to hand this off to you and let you take it away.
Hello, everyone. Come on in. Welcome to our multi-user space. As Cinnamon said, my name is Debbie Steele. I am a faculty research assistant in the Cetacean Conservation Genomics Lab, which is part of Oregon State University and the Marine Mammal Institute, and we're based here at the Hatfield Marine Science Center. Forgot what I was going to say next already. Now that, that's all quite a mouthful, but what it basically means is that I'm a researcher in a group that uses DNA to answer questions about whales and dolphins. So now that you've heard me speak for a little bit, you're probably thinking to yourself, she doesn't sound like she's from the US, and you would be right. As Cinnamon mentioned, I am from New Zealand originally, and despite living here for about 15.5 years now, my accent has not softened all that much, I'm told. So with that in mind, if I say anything that you don't understand, either because of my accent or for any other reason, or if I mention something that you want to hear a little bit more about, please type into the chat and Cinnamon will let me know and I can hopefully answer your question. All right, with that out of the way, today I'd like to talk to you a little bit about DNA, what it is, how we get our little hands on it, and a couple of things that we do with it once we have it. DNA stands for deoxyribonucleic acid. It is found in the cells of pretty much every living thing on the planet. It is made up of just 4 smaller molecules called bases, A, G, C, and T. And those are joined together in long strings. It is the order and grouping of those bases that contains all the information a thing needs to function, basically. And in general, the smaller and more simple an animal or plant is, the less information it needs. So like this basic Lego model here, or like a virus, for example, not a lot of instructions needed to run that organism. And just speaking of viruses, the virus that causes COVID has a small instruction manual of about 30,000 bases. So 30,000 of those A, G, C's, and T's strung together. When you move to more complex things like humans, or like this bigger Lego set here, you need a lot more instructions for us to function properly. And humans come in at about 3.2 billion bases. So that's about 100,000 times as large as that virus genome that I was talking about. So that's pretty much DNA in a nutshell. Is there any questions about that before I move on to how we get our beady little hands on it? All right, so If any of you have had a COVID test in the last couple of years, you will be very familiar with these things. This is what gets kind of shoved up your nose and rolled around to take a sample to check whether you're infected with the virus. And this is a pretty simple technique for collecting a sample. Simple, somewhat uncomfortable technique, I will say. But it's probably not one that we can translate to using with whales and dolphins. I don't know many of them that are going to just hang around and let us stick one of these down their blowhole. So we need to come up with different ways to get a sample from them. And actually, there's a whole heap of them, and I'm just going to talk about 4 different ways today. The first we get using one of these. This is fired from a crossbow, and this end down here, this metal tip, will then hit into the side of a whale, bounce back out, and in it will be a small, we call it a biopsy sample. It's a small plug of tissue, skin and blubber, and then the dart floats in the water and researchers can come along, pick it up. And the sample we get from that looks a little bit like this. Can you see that? Get my thumbnail out of the way. So hopefully you can see, it might be a little bit cloudy from the ice condensation, but hopefully you can see some black skin and then a little white layer of blubber there. This biopsy sample is the most common sample that we deal with in this lab. And it's the type of sample that gives us the best quality DNA and the most of it. The absence of one of these, we have a few other things that we can do.
Before you go on, Debbie,
yeah,
can you show us that crossbow dart one more time?
Sure. I can model it. Do you want to see the end?
Great.
Good.
Thank you.
So whales and dolphins are like humans in that they shed their dead skin. So we can also scoop that out of the water, and that's what this little fleck in here is. So hopefully you can see that little black piece of skin in the tube. That is some dead skin that's sloughed off a humpback whale in Glacier Bay. The third sample I want to talk about is something else we can scoop from the water, and that is fecal matter or whale poop. And that is what you can hopefully see in the bottom of here. All of that kind of black gunky stuff there is whale poop.
So
the
Debbie,
bonus,
we're having a couple of issues with folks that are watching from a different kind of platform. Can you have Mark see that real quick and then zoom in so that Yicheng can see it? So that way folks, depending on what they're viewing, can see it, okay, and then go back to Yicheng. All
right, so that's it in Mark's camera. And that's it in Yicheng's camera.
Perfect, thank you.
No worries. So the bonus with this kind of sample is not only can we use it to get the DNA of the whale that did the poop, but we can also get the DNA of what the whale's been eating. So that's kind of cool. And then the last sample I wanted to talk about, and hopefully Cinnamon can play a little video of how this is collected, is whale blow. And so I assume what you're watching right now, because I can't see.
It's okay, hang on, give me a second to get it started.
Just let me know when it's going.
All right, everybody.
Yeah, we're good. Okay, so what you're seeing is a drone flying over the top of a blue whale. And on that drone, you can see some plastic petri dishes. And so hopefully Cinnamon will play that again. When the whale comes up to the surface to expel its air or blow out, that drone will collect that blow, that whale snot. And these drones have been affectionately termed snot bots in those petri dishes, and we can get DNA from the snot of a whale as well. So with all, now I'm looking at myself, with all those different tissue types, we use slightly different methods to extract DNA from each of them. And I'm not really going to go into the details of how that's done today, other than to say each of those methods will use a combination of both mechanical and chemical processes to basically take the DNA out from all of the other gunk that exists in the sample around it, all the fats, the proteins, seawater, anything else that's there. And at the end of the day, once you've gone through all of that, you have an exciting tube of clear liquid like this. And this is DNA suspended in a buffer. which helps keep it safe and can survive for a long time in the freezer, so we can use it over and over again. So I'll get that in front of this camera as well. All right, and so that's basically how we get it and the different samples we use. And so I don't know, Cinnamon, if there are any questions about that. Maybe I'm speaking too fast in my New Zealand accent.
What kind of whale was that first sample taken from?
The very first biopsy sample?
Yeah.
This is from a humpback whale in Mexico in 2004.
Nice. And how about the poop sample?
The poop sample, wow, that's kind of cool. I'll get that back out as well. This was collected by one of the other research groups in the Marine Mammal Institute, the GEM Lab, which I believe stands for Geospatial Ecology of Marine Mammals. And they have a project off the coast looking at the grey whales, the Pacific Coast Feeding Group grey whales. And they're a set of whales that, instead of migrating all the way up to the Arctic to feed, actually hang off the coast here in Oregon. And they collected these poop samples. And one of the master's students in our group is actually looking at these to try to work out what prey the grey whales have been eating. So,
can you tell from your snot and poop samples if the whales are healthy?
You can actually, because that's one of the other things that GEM Lab is looking at with the poop samples, is they're doing hormone extractions. And so they can look at things like stress hormones and stuff like that to just, to see how the whale's doing. And same with the blow samples that are collected. We get some of those samples for DNA, and there's another research group that takes those and does hormone extractions on those as well. So, yep.
And how is fecal samples or poop samples collected from a whale?
I think with a lot of perseverance and luck. I don't actually go out on the water, but Lee tells me that it can take some time. They follow a whale around and just kind of hope that at some point it's going to release its fecal matter. They do send up drones, which help. And then when the whale poops, they speed over in their boat and use a kind of a plankton net, I believe, to scoop that out of the water and then rinse it off from the net into tubes. So, and that's just a subsample because, like I said, they use it for hormones. So we just get a small sample. There's usually a lot more, as you can imagine. Whales don't do small poops.
Great. Thank you so much. Questions are still coming in. Keep typing them in, everybody. And Debbie, you can move to your next thing.
All right. So we're actually literally going to move now. As I mentioned, this is a multi-user lab space. We are one of 3 genetics groups that are based out here at Hatfield. The other 2 are mostly doing fisheries genetics research. And we have our own kind of separate areas out here in the main lab, which I believe Ichung is showing you. And then off to the side, we have these specialty use rooms, which we share. And the room we're about to enter now, we call the equipment room. So come on in. And this is Charles, the master's student I was mentioning earlier, who's doing the fecal analysis. So we'll get to what he's doing in a little bit, but we're going to start over here with some of our PCR machines. And so PCR may be a term that's familiar to some of you. You would have heard it with regards to COVID testing. It's one of the 2 types of tests that we can do to see if someone's infected with COVID. PCR stands for polymerase chain reaction. And at its basics, it's essentially just using an enzyme, a polymerase, to make heaps and heaps of copies of a certain small fragment of DNA. And that's what's going on over here. We've got a PCR cycling. One of the things that we can use PCR for is to genetically sex a whale or dolphin. And this is useful because for a lot of species, you can't actually tell whether it's a male or a female just by looking at it. And so we target actually 2 different parts of the DNA to do this test. We have a 200 base pair fragment on the Y chromosome that's only found in males, and then about a 400 base pair fragment on the X chromosome, or that's found in both males and females. And that size difference really matters because once we've done our PCR, the way we can tell our results from that amplification or that making of heaps and heaps of copies is by running it out on a gel. And so we're going to flip around and see what Charles is doing over here, because he is loading the gel. And if you zoom in there and see. So what he's doing is in each of these wells here, sorry, Charles, interrupt your work. He is loading PCR product. Like you can tell from the DNA tube I showed you earlier, we deal with a lot of clear liquids. So he's actually using this blue dye to help him see what he's doing as he puts the DNA into each of these wells. And you can kind of think of this gel as like a obstacle course for DNA. It's a pretty tight matrix set in there. He's putting the PCR, the DNA, into those wells. DNA is negatively charged, and when he sets this going, which it looks like he's about to do, he's going to set up an electrical field across the gel, and the DNA is going to be attracted to the positive end of that gel, kind of in the same way that different ends of a magnet want to join together. So the DNA is going to want to push its way through that gel to get to the positive end. And as you can probably imagine, the smaller the DNA fragment, the easier it's going to be able to wiggle through that gel and get to the other end. So once we've run it for a while, the smaller fragments will have moved further than the larger fragments, and we'll be able to see our two different sized bands that we amplified in the PCR, the male band and our control band. And we can do that by using this machine over here, which is our gel dock. This shines a UV light on the gel, and that fluoresces the stain that we've applied to the gel, and that gives us a lovely gel image like this. And so hopefully you can see these dark holes up here. These are the wells that Charles was just loading into. This is the negative end, and the PCR product has run this way through the gel. On the right here, we can see a ladder. Each of these bands is 100 base pairs different in length. So we've got 100, 200, 300. And then each one of these lanes is a different whale. And so you can see these ones with the two bands, they're males. And then if we look down here, we can see some that only have one band, and those are females. So Cinnamon, any questions about sex PCRs or genetic sexing of whales and dolphins before we continue?
We don't have any questions at this time, but I did want to let folks know that are watching the Q&A that Scott Baker, the researcher that works with this lab, is also answering some of your questions. So if you have a question we haven't answered, you can watch for Scott's response. And that might help get to some of these that we're not able to get to while we're there. But go ahead and continue, Debbie.
All right, so the next thing I was going to move on to is one of the other things that we can use PCR for, which is to genetically identify species of whale or dolphin. And so you might just be sitting there thinking, why on earth would I need to use DNA to do something like that? I see a killer whale, it looks like a killer whale. I see a blue whale, it looks like a blue whale. And you are absolutely right. Those ones are pretty easy. I think even I, who doesn't go out in the field, could identify those. However, any of you that were here for Dr. Lisa Balance's talk earlier this morning will have had a preview of what I'm about to talk about next. There is an entire group of cetacean that does not make it easy for us to identify in the field for a number of reasons. And that group is called the beaked whales. And if you want to hear more about the beaked whales and you didn't listen to Lisa's talk this morning, I encourage you to tune into the recording at some point. I'm just going to touch on them briefly here today.
One quick question, Debbie,
yeah.
Before we go to that. Do you make your own primers?
We do not make our own primers here. I order them from a company and they make them and send them out to me.
Okay,
that's
great.
A good question.
Thank you.
Someone who knows some stuff. So yeah, beaked whales, they like to live out in the open ocean. They like to dive deep. They spend a lot of time under the water. You got to be pretty lucky to see them at the surface. And even if you do see them, they have this additional nasty habit of looking a lot like each other. So it's oftentimes you're just looking at this gray beaked looking thing in the water and you're not sure what kind of species it is. And that's exactly what happened last year when the Marine Mammal Institute researchers went on a cruise just off the Oregon coast, and they came across this group of beaked whales that hung out around their boat for quite a while. They managed to get audio recordings, a lot of photographs, videos, and they collected a biopsy sample. And that got brought back here. And we went through that whole process I described earlier. In fact, the DNA tube I showed you earlier was the DNA from that beaked whale. We got its DNA. We went through the PCR. We were able to determine that the biopsy sample they collected came from a female. And then we also amplified a different region and then put it through kind of a modified PCR after that, which instead of making heaps and heaps of copies of the entire fragment of DNA, we make copies of every different size within that fragment. So then we can use the machine behind you to look at that. And so inside this machine, we're going to have to do a little dance here. Inside this machine, well, this is a DNA analyzer, fragment analyzer. We use it to do both sequencing and something I'm not going to talk about today, but individual identification as well of whales and dolphins. Inside the guts of this machine is, I'm going to switch with you, Ishong, is this, which I have sitting safely here on the table, because this is a brand new one of these. Usually we have old ones left around for demonstrations like this, but as Cinnamon alluded to, we moved into this building last year, and during that move, we threw out all of our handy dandy props. So we have to use this brand new one. And I'm a little nervous to touch it because these things are not cheap. So if you want to take a minute to just type into the chat your guess on how much you think this costs, I will give you a hint. It's less than $10,000. Charles, you can also guess if you want. And this is I'm going to have to try to say the word properly. This is a capillary array. I keep wanting to say capillary. There are 96 tiny little tubes here, and inside each of these tiny little tubes is a gel matrix similar to the one I described before, so that DNA obstacle course. And hopefully you can see this kind of roller coaster ride that the DNA is going to go on. It gets sucked up at this end and then travels up and around and down through the capillaries to get to this little window. And the distance that it travels through those is a lot further than on that agarose gel we saw earlier. And that extra distance allows us to separate the fragments out a lot more. And so what finally comes past that window are DNA fragments that are one base pair different in size. So Cinnamon, were there any guesses on how much this cost?
Oh, there were lots of guesses and they run the gamut. Do you want to tell us the actual answer?
Well, anyone who got between 6,500 and 7,000 is close enough, I would say. It is $6,730 was what this cost.
Nice. We have some that are very close. Nice job, everybody.
Nice. And this usually lasts for about, I think Dave said 1,000 runs. So that comes in at around $7 a run just for the array, let alone everything else that goes into this machine. Anyway, so we get down to this little window here where you can see all of these capillaries come quite close together. And inside the machine, aimed at that window, is a laser. And that laser will excite a dye that we've attached to each of those different fragments. And the color of that dye depends on what base was at the end of that fragment. So if I'm remembering rightly, I think it's a red if it's a T, it's yellow if it's a G, green if it's an A, and blue if it's a C. Don't quote me. And then that dye fluoresces, it gives off a burst of light, and with the laser there's a camera that captures that fluorescence. And then we get a picture like this on the screen over here, which we're getting to slowly. There we go. And so each one of these lanes heading up is one of those capillaries. So there are 96 samples across this. This is, I believe, the short end, and this is the long end. So the DNA has run this way. And each of these different colored bands is one of those bases fluorescing. So you can look at that and you could probably go through and work out manually, if you wanted to zoom in, what the sequence was. Thankfully, we have computers to do our work for us. So I'm going to switch to this and just bring up, I'm going to share my screen, Cinnamon.
Looks good, Debbie.
Okay, and this is what it looks like. And this is the actual sequence that we got from that beaked whale sample that was collected last year off the coast of Oregon. And so you can see each of these different colored peaks. We'll see if I guessed right what the colors of each of the bases were. So you can see my mouse, right, Cinnamon?
Yes, we can see your mouse.
Yeah, so here's a T followed by an A, followed by a C, followed by another A. And you can see each of these peaks is essentially that burst of light coming at the camera. And then across the top, the software has called all of these bases for us, depending on what color each of those peaks were. So that makes it a lot easier. We can then take that sequence and put it into our curated reference database, which contains sequences from almost all, I could used to say all, but we keep getting new species described. So almost all known species of whales and dolphin. And we can, that's called Witness for the Whales. And it is a web-based program. So if you wanted to go and have a play with it, you could. What we do is paste those A, G, Cs, and Ts that I described earlier into this little box here. We run a simple cluster analysis and select the database that we want to run our sequence against. And I know that the sample came from a beaked whale, so I'm just going to go straight to the beaked whale family, which is called Ziphidae, and that is why I've selected this database here. You click submit, and after a little bit, it comes back with a tree. And these are almost all the beaked whale species that we know about, and a few samples from each of those. The whole tree is over there on the left, and I've kind of just zoomed into the portion of interest here that we have on the right. And you can see here is our little sample sitting down in this group of Hubb's beaked whales. And in fact, when we look at the sequence more closely, we can see that it's identical to this other known specimen of Hubb's beaked whale. So we are 100% sure that the biopsy sample that was collected came from a Hubb's beaked whale. And just to bring it all back to COVID, if you are looking at this tree and thinking, you know, I've kind of seen something like that before, you might be thinking about some of these that have appeared in the media. I just grabbed these off the web in the last few days. And this is a similar kind of concept to what I was showing you before with the whales. It's just a way of grouping similar sequence types together and displaying them visually. And each of the clusters here is a different variant that they've sequenced of the COVID virus. So that is us. And so I don't know if there's any questions that you have there, Cinnamon. I think we've still got a couple of minutes.
We do. Will you stop sharing your screen for us?
I will.
We can focus on you. For everybody online, keep those questions coming. Scott Baker's been doing a really great job of answering some of your more detailed questions. So we appreciate that. Let's see. So question that I was curious about, Debbie, while we're waiting for folks to add some more questions in our last couple minutes, can you talk about the completion of the human genome and what was just done?
Oh, yeah, sure. So that came as a bit of a surprise to me, actually, because I honestly thought that they'd finished that a few years ago. So when I read that news article, I was like, there you go. So yes, they finally finished off the last few parts of the human genome, I believe. So what I didn't really go into when I described DNA is that it's not just as simple as a simple string. It gets bundled up and packaged up and arranged in different ways. And so there's kind of some tricky parts of that DNA to get into, to break into, to do that sequencing. And I think it was some of those final tricky parts that they just managed to finish off in the last few months, I guess. So yeah, I believe now it is officially fully done.
Great. And we had a question come in about what do you do with a sample that doesn't match any of those already on record when you were showing us the tree?
Well, that is a really good question. And that actually did happen to me. So when I first started working for Scott, gosh, almost 20 years ago now, we had a sample sent to us from one of the Pacific Islands, and they didn't know what kind of whale or dolphin this had come from. So I went through that process that I described before, put it into the tree and went, this looks a bit weird because it's kind of sitting near ginkgo tooth beak whales, but it's not really close enough to be a ginkgo tooth. And I didn't back then really know all that much about whales and dolphin genetics. So I went to Scott and showed him, and he also thought it looked a little bit weird. And as it turns out, that was a new species of beaked whale. I am not going to try and pronounce its common name because I'm not very good at that, but hopefully Scott can type that in and you can see what it is. Um, so yeah, that was kind of fun and exciting.
Always great to learn new discoveries. We have
Yep.
hit our time block, Debbie, and I just want to say thank you so much for taking us through your lab. For everybody online, thank you so much for working with us as we work through all this technology. Thank you to Scott Baker online for helping with questions. Thank you to E. Chung Chung and to Mark Farley, our wonderful photographers and videographers that are working in the room. For folks online, you have a few minutes before you can go back and watch the next a live animal interaction. The Sea Grant folks are going to be talking about sea cucumbers. So if you're interested in that, Shannon, will you put into the chat or Q&A the link where folks can get back to the live events so they can get to their chat, get to their event. And for everybody else, if you're not interested in learning about cucumbers, everybody is, but if you'd like to spend your time somewhere else, please explore the website. There's lots of things to see and do there, and enjoy Marine Science Day. Thanks, everybody. We'll see you again somewhere else in this virtual world. Debbie, thank you so much. We appreciate it.
Thank you. Bye.